Imagine looking back through time, not just years or centuries, but billions of years into the past. Picture our planet as it was 4.5 billion years ago, a molten hellscape where rocks fell from the sky like rain, where the surface was so hot that metals would flow like water, and where the very air would kill any life form we know today in seconds. This was Earth before life, a world that seemed absolutely hostile to everything we consider living. Yet somehow, in this impossible environment, something extraordinary happened. The first spark of life appeared. But how? How did lifeless chemicals transform into something that could grow, reproduce, and eventually evolve into every living thing we see around us today? This is perhaps the greatest mystery in all of science. It's a question that has puzzled humans for thousands of years, and one that we're still trying to fully answer today. Welcome to the story of life's beginning, a journey that will take us from the chemistry of dying stars to the first cells that would change our planet forever. Before we dive into the deep past, we need to ask ourselves a fundamental question. What exactly is life? It might seem obvious at first. You know life when you see it, right? A dog is alive, a rock is not. A tree is alive, a car is not. But when scientists try to study how life began, this simple question becomes incredibly complicated. Think about it for a moment. What makes something alive? Is it movement? But rivers move, and they're not alive. Is it growth? Crystals grow, but we don't consider them living. Is it the ability to reproduce? Some living things, like mules, cannot reproduce. Is it having DNA? But viruses have DNA, and scientists debate whether they're truly alive. The more we examine life, the blurrier the line becomes. Scientists have proposed many definitions over the years. Some say life is anything that can grow, reproduce, and respond to its environment. Others focus on metabolism, the chemical processes that keep an organism running. Still others point to evolution, the ability to change over generations through natural selection. But here's where it gets really interesting. When we're looking for the origins of life, we're looking for the moment when non-living chemistry became living biology. That means we need to understand exactly where to draw that line. Did life begin with the first self-replicating molecule, the first cell with a membrane, the first organism that could evolve? This isn't just an academic debate. The definition we choose shapes everything about how we search for life's beginnings. If we define life as anything with DNA, then we're looking for the origin of genetic systems. If we define it as anything that metabolizes, then we're searching for the first chemical reactions that could sustain themselves. For our journey through the origins of life, we'll use a working definition that captures what most scientists agree on. Life is a system that can maintain itself through metabolism, reproduce itself through some form of heredity, and evolve through variation and selection. This definition helps us focus on the key transitions that had to happen for life to emerge from non-life. But even with this definition, we face another challenge. Life today is incredibly complex. Even the simplest bacteria have thousands of different proteins, complex genetic systems, and sophisticated molecular machinery. How did we get from simple chemicals to something so intricate? It's like trying to understand how a Boeing 747 was built by studying only the finished airplane, without the blueprint, the factory, or any of the intermediate steps. This is why the origin of life is such a fascinating puzzle. We have to work backward from the complex life we see today, piece together clues from chemistry and geology, and try to imagine what the simplest possible form of life might have looked like. We have to think about what came before cells, before DNA, before proteins, all the way back to the basic chemistry that made everything else possible. As we embark on this journey, remember that we're not just exploring the past. Understanding how life began helps us understand what life truly is, what makes our planet special, and whether life might exist elsewhere in the universe. The answers we find don't just tell us where we came from, they help us understand where we might be going. To understand how life began on Earth, we first need to understand what Earth was like when it formed. The period we're talking about is called the Hadean Eon, named after Hades, the ancient Greek underworld. It's a fitting name because early Earth was quite literally hell. Picture this. 4.5 billion years ago, our solar system was a chaotic nursery of forming planets. Earth was being bombarded constantly by asteroids, comets, and other chunks of rock and ice left over from the solar system's formation. These impacts were so frequent and so violent that they kept the entire surface of our planet molten. Imagine an ocean made not of water, but of liquid rock, glowing red-hot and stretching as far as the eye could see. The atmosphere, if you could call it that, was nothing like what we breathe today. There was virtually no oxygen. That would come much later. Instead, the air was filled with water vapor, carbon dioxide, methane, ammonia, and hydrogen sulfide. The pressure was crushing, and the temperature was hot enough to melt lead. Any water that tried to exist as a liquid would instantly boil away into steam. But here's the remarkable thing about this hellish landscape. It was being constantly enriched with the raw materials of life. Every asteroid that crashed into Earth brought with it chemicals forged in the hearts of dying stars. Carbon, nitrogen, phosphorus, sulfur. All the elements that would eventually become part of living things were being delivered to our planet in a cosmic bombardment that lasted for millions of years. These elements had been created in stellar furnaces billions of years earlier. When massive stars reached the end of their lives and exploded as supernovas, they scattered these heavy elements throughout space. Some of them eventually found their way into the cloud of gas and dust that would become our solar system, and from there into the planets themselves. So even in this lifeless hellscape, the chemistry of life was being assembled. Iron for carrying oxygen and blood, calcium for building shells and bones, phosphorus for storing energy and information in DNA. All of it was there, waiting in that molten ocean for conditions to change. And change they did, though slowly. As the bombardment began to ease up after about 500 million years, the surface of Earth started to cool. The molten rock ocean began to solidify into the first solid crust. Water vapor in the atmosphere started to condense, and for the first time in Earth's history, rain began to fall. But this wasn't gentle spring rain. This was a torrential downpour that lasted for millions of years. The rain fell so hard, and for so long that it carved out the first river valleys and filled the first ocean basins. The water that fell was scalding hot, probably close to the boiling point, but it was liquid water nonetheless. For the first time, Earth had oceans. These early oceans were very different from the ones we know today. They were hot, acidic, and filled with dissolved minerals from the constant erosion of the rocky crust. They contained virtually no oxygen, but they were rich in hydrogen, methane, and other simple gases. Most importantly, they provided a stable environment where complex chemistry could begin to happen. The water itself was crucial because it could dissolve and transport chemicals in ways that wouldn't be possible on a dry world. It could bring different molecules together, allow them to react, and carry away the products of those reactions. Water also provided a medium where large, complex molecules could fold into specific shapes, something that would prove essential for life. As the oceans stabilized, something remarkable began to happen in their depths. The same chemical processes that had been creating simple organic compounds in space began to occur on Earth. Lightning strikes in the atmosphere created complex molecules that rained down into the oceans. Volcanic vents on the ocean floor provided energy for chemical reactions. The stage was being set for something unprecedented in the universe, the emergence of life from non-life. Before we dive into the real science of how life began, it's worth taking a moment to understand how humans have thought about this question throughout history. For most of human civilization, people believed that life could spontaneously emerge from non-living matter all the time. This idea, called spontaneous generation, seemed obvious based on everyday observations. Aristotle, the great ancient Greek philosopher, was one of the most influential proponents of spontaneous generation. He observed that worms appeared in mud after rain, that maggots emerged from rotting meat, and that mice seemed to generate spontaneously from grain left in dark corners. To Aristotle and his contemporaries, it was clear that life could arise directly from the right combination of matter and conditions. This idea persisted for over 2,000 years. Medieval scholars believed that mice could be created by leaving dirty rags in a dark corner with some grain. They thought that fish could spontaneously appear in ponds, and that insects emerged directly from decaying matter. even respected scientists, as late as the 17th century believed in some form of spontaneous generation. The problem was that these early thinkers didn't have the tools to see what was really happening. They couldn't see the tiny eggs that flies laid on meat, which later hatched into maggots. They couldn't observe the microscopic organisms that were actually responsible for decay and fermentation. They couldn't detect the seeds and spores that gave rise to new plants and fungi in seemingly sterile environments. It wasn't until the invention of the microscope in the 17th century that scientists began to realize how complex and interconnected life really was. Even then, the debate continued. When Antony van Leeuwenhoek first observed bacteria and other microorganisms, many scientists assumed these tiny creatures were spontaneously generating in the water. The death blow to spontaneous generation came, in the 19th century, through the careful experiments of scientists like Louis Pasteur. Pasteur showed definitively that life does not spontaneously arise from non-living matter under current Earth conditions. His famous swan neck flask experiments proved that even bacteria and other microorganisms come from pre-existing life. When he sterilized broth and protected it from contamination, no life appeared. But when he allowed microorganisms to enter the broth, life flourished. Pasteur's work established one of the fundamental principles of modern biology. Life comes from life. This principle, known as biogenesis, tells us that all living things arise from other living things through reproduction. It was a revolutionary idea that helped establish biology as a rigorous science. But Pasteur's discovery also created a paradox that we're still grappling with today. If life can only come from other life, then how did the first life begin? This question became known as the paradox of biogenesis. It forced scientists to realize that the origin of life must have been a unique event in Earth's history, something that happened once under very special conditions that no longer exist. This is why studying the origin of life is so different from studying any other biological process. We can't observe it happening now because the conditions that allowed it to happen no longer exist on Earth. We have to reconstruct those ancient conditions and figure out what kinds of chemistry might have been possible billions of years ago. That isn't possible today. The key insight is that while spontaneous generation doesn't happen under current conditions, it might have been possible under the very different conditions of early Earth. The chemistry that gave rise to the first life was probably nothing like the spontaneous generation that Aristotle imagined. Instead, it was likely a gradual process that took millions of years and required a very specific set of environmental conditions. This realization opened up an entirely new field of scientific inquiry. Instead of looking for life appearing spontaneously from non-life, scientists began to search for the chemical pathways that could have led from simple organic molecules to the first self-replicating systems. They began to study what kinds of chemistry might have been possible in Earth's early oceans, atmosphere, and crust. The journey from Aristotle's worms to modern origin of life research shows us how science progresses. Sometimes what seems obvious turns out to be wrong, and the real explanation is far more subtle and interesting than what we first imagined. The origin of life isn't about worms appearing in mud. It's about chemistry so elegant and complex that it took scientists hundreds of years just to begin to understand it. In 1953, a young graduate student named Stanley Miller walked into his advisor's laboratory at the University of Chicago with an ambitious idea. He wanted to recreate the conditions of early Earth and see if he could make the building blocks of life from scratch. His advisor, Harold Urey, was skeptical, but agreed to let Miller try. Miller built a simple but elegant apparatus. He filled a sealed glass container with water to represent the early oceans, and above it he put a mixture of gases that they believed made up Earth's early atmosphere: methane, ammonia, hydrogen, and water vapor. Then, he ran electrical sparks through the gas mixture to simulate lightning. After just 1 week, Miller had created something remarkable. The clear water in his apparatus had turned a murky brown, and when he analyzed what was in it, he found amino acids, the building blocks of proteins. Not just 1 or 2 amino acids, but many of the same ones that are essential for life today. Miller had shown that the basic chemistry of life could emerge naturally from simple starting materials. This experiment launched the idea that became known as the primordial soup hypothesis. The idea was that early Earth's oceans gradually filled up with organic molecules created by lightning, volcanic activity, and other energy sources. Over time, this soup became richer and more complex until somewhere in its depths, the first living things emerged. The primordial soup became one of the most famous ideas in origin of life research. It was elegant, easy to understand, and seemed to solve the basic problem of how you get complex organic molecules on a lifeless planet. Textbooks around the world began teaching students that life emerged from a soup of organic chemicals in the early oceans. But as scientists learned more about early Earth and about the chemistry of life, they began to find problems with the simple soup idea. The first issue was with the atmosphere Miller had used in his experiment. New evidence suggested that early Earth's atmosphere was probably not as rich in hydrogen, methane, and ammonia as Miller had assumed. Instead, it was likely dominated by carbon dioxide and nitrogen, gases that don't produce organic molecules as easily when you run electricity through them. When scientists repeated Miller's experiment with this more realistic atmosphere, they got very different results. Instead of a rich variety of amino acids, they found mostly simple compounds that weren't particularly useful for building life. The soup seemed to be getting thinner. An even bigger problem emerged when scientists began to think more carefully about what would happen to organic molecules in the early oceans. It turns out that the same conditions that create organic molecules, high temperatures, electrical discharges, ultraviolet radiation, also tend to destroy them. The molecules that Miller created in his flask would have quickly broken down if they were exposed to the harsh conditions of early Earth. There was also the concentration problem. Even if organic molecules could survive in the early oceans, they would have been incredibly dilute, scattered throughout vast volumes of water. Getting them concentrated enough to interact and form more complex structures would have been extremely difficult. It would be like trying to make a cake by throwing flour, eggs, and sugar into a swimming pool and hoping they would somehow find each other. These problems led many scientists to question whether the primordial soup ever really existed. Maybe the early oceans weren't filled with organic molecules after all. Maybe life didn't begin in some warm little pond, as Charles Darwin had once suggested, but somewhere else entirely. But here's the interesting thing about science. When one idea doesn't work out, it often leads to better ideas. The problems with the primordial soup hypothesis forced scientists to think more creatively about where and how life might have begun. If not in a soup scattered throughout the oceans, then where? Some scientists began to focus on specific environments where organic molecules might naturally concentrate. Tide pools, for example, could concentrate molecules as water evaporated. Clay minerals could absorb and organize organic compounds on their surfaces. Ice could trap molecules in concentrated pockets of liquid water. Others began to think about whether life might have begun not with a soup of disconnected molecules, but with simple chemical systems that could organize and sustain themselves. Maybe the key wasn't having lots of organic molecules floating around, but having the right kind of chemistry in the right place at the right time. The debate over the primordial soup teaches us something important about how science works. Miller's experiment was brilliant and groundbreaking, and it opened up an entire field of research. But science doesn't stop with one experiment or one idea. Scientists keep testing, questioning, and refining their understanding. Sometimes this means abandoning ideas that once seemed promising, but it always leads to a deeper and more accurate understanding of how the world works. Today, most scientists don't think life began in a simple primordial soup, but Miller's legacy lives on in the sophisticated experiments that scientists now use to study the origin of life. His basic insight, that you can recreate the conditions of early Earth in the laboratory and see what kind of chemistry emerges, remains the foundation of origin of life research. As scientists moved beyond the simple primordial soup idea, they ran headfirst into one of the most fundamental puzzles in all of biology. It's a puzzle that goes to the heart of how life works today, and it makes the origin of life seem almost impossibly difficult to explain. Here's the problem. In every living cell on Earth today, there are two types of molecules that depend completely on each other. Proteins do almost all the actual work in cells. They speed up chemical reactions, transport materials, provide structure, and carry out the countless tasks that keep life running. But proteins can't make copies of themselves. For that, cells need DNA. DNA stores the information that tells cells how to make proteins. It's like a blueprint or instruction manual written in a 4-letter chemical code. But here's the catch. DNA can't do anything by itself. To read the information in DNA and use it to make proteins, you need other proteins called enzymes. Do you see the paradox? Proteins need DNA to tell them how to be made, but DNA needs proteins to be read and used. It's like having a book that can only be read by someone who already knows what the book says, or a factory that can only be built by workers who can only be trained in that same factory. This is sometimes called the chicken and egg problem of molecular biology. Which came first, proteins or DNA? How could either one exist without the other? And if both are required for life as we know it, how could life ever get started? When scientists first discovered this paradox in the mid-20th century, it seemed like it might make the origin of life impossible to explain scientifically. Some researchers even suggested that life must have been created by an intelligence of some kind, because the interdependence of proteins and DNA seemed too complex to have emerged naturally. But other scientists weren't ready to give up. They began to look more closely at the chemistry of proteins and DNA to see if they could find clues about how this system might have evolved. What they discovered was that while proteins and DNA are incredibly sophisticated, they're both made up of much simpler building blocks. Proteins are chains of amino acids linked together in specific sequences. There are 20 different amino acids used in life, and they can be combined in countless ways to make proteins with different shapes and functions. The key insight was that some very simple chains of amino acids can have useful properties even if they're nothing like the complex proteins found in modern cells. DNA is made up of 4 different chemical units called nucleotides, linked together in long chains. Each nucleotide consists of a sugar, a phosphate group, and one of 4 bases, A, T, G, or C. The sequence of these bases spells out the genetic code, but individual nucleotides and short chains of nucleotides can also have interesting chemical properties. This led scientists to wonder whether there might have been simpler versions of proteins and DNA in the early stages of life's evolution. Maybe life began not with the sophisticated molecular machinery we see today, but with much cruder versions that could gradually be improved over time. Some researchers began to focus on the possibility that very simple proteins might have been able to catalyze their own formation or the formation of simple genetic molecules. Others explored whether simple genetic molecules might have been able to direct the formation of basic proteins. But even these simplified versions of the chicken and egg problem seemed difficult to solve. Simple proteins still needed some way to store and pass on information about their structure. Simple genetic molecules still needed some way to influence the chemistry around them. The breakthrough came when scientists realized they might have been thinking about the problem in the wrong way. They had been assuming that proteins and DNA were the only possibilities for the first biological molecules. But what if life began with something else entirely? Something that could both store information like DNA and speed up chemical reactions, like proteins? This insight led to the discovery of one of the most important concepts in origin of life research: the idea that there might have been a simpler form of life that existed before the protein-DNA system we see today. This hypothetical early life would have been based on molecules that could do double duty, acting as both information storage and catalysts. The search was on for molecules that might have these dual capabilities. Scientists began to look at the chemistry of various organic compounds to see if any of them might be able to both carry information and speed up chemical reactions. What they found would revolutionize our understanding of how life might have begun. The paradox of proteins and DNA taught scientists an important lesson about studying the origin of life. Sometimes the biggest obstacles to understanding can lead to the biggest breakthroughs. By forcing researchers to think outside the box of modern biochemistry, this paradox opened up entirely new ways of thinking about what the first life might have looked like. In the 1960s and 70s, as scientists grappled with the chicken and egg problem of proteins and DNA, a few researchers began to take a closer look at a molecule that seemed to be playing a supporting role in the story of life. RNA, or ribonucleic acid, is DNA's close chemical cousin. Like DNA, it's made up of 4 different nucleotides linked together in chains, but there are some key differences. While DNA uses the bases A, T, G, and C, RNA uses A, U, G, and C, substituting uracil for thymine. RNA is also usually single-stranded, rather than double-stranded, and it uses a slightly different sugar in its backbone. In modern cells, RNA plays several important roles. It carries information from DNA to the protein-making machinery of the cell. It helps translate the genetic code into proteins. It's even part of the molecular machines that actually build proteins. But for most of the 20th century, RNA was seen as just a messenger, a molecule that carried information but didn't do much else. That began to change when scientists discovered that some RNA molecules could do something remarkable. They could act as enzymes. These special RNAs, called ribozymes, could speed up chemical reactions just like protein enzymes. This was revolutionary because it suggested that RNA wasn't just a passive carrier of information, it was an active participant in the chemistry of life. The discovery of ribozymes sparked an exciting new idea. What if early life was based entirely on RNA? What if there was once an RNA world, where RNA molecules served both as genetic material and as enzymes? This would solve the chicken and egg problem in one stroke. RNA could store information like DNA and catalyze reactions like proteins. The more scientists thought about this idea, the more attractive it became. RNA has all the right properties to be the basis for early life. It can store information in its sequence of bases, just like DNA. It can fold into complex three-dimensional shapes that can catalyze chemical reactions, just like proteins. And most importantly, it can make copies of itself under the right conditions. The laboratory, scientists began to demonstrate just how versatile RNA could be. They found ribozymes that could cut and paste RNA molecules, add nucleotides to growing RNA chains, and even evolve new functions through random mutations and selection. Some ribozymes could catalyze reactions involved in metabolism. Others could help build the very nucleotides they were made of. The RNA world hypothesis became one of the leading theories for the origin of life. The idea was that somewhere on early Earth, perhaps in tidal pools or around hydrothermal vents, the first self-replicating RNA molecules emerged. These primitive ribozymes could make copies of themselves, and any that were better at replication would become more common over time. As these RNA populations evolved, they would have developed more sophisticated capabilities. Some RNAs might have specialized in storing information, while others focused on catalyzing useful chemical reactions. Eventually, the system might have evolved to the point where it could begin making proteins and DNA, gradually transitioning to the dual molecule system we see in life today. The RNA world hypothesis was elegant and compelling, but it also faced some significant challenges. One of the biggest problems was the question of how the first RNA molecules could have formed on early Earth. RNA is a complex molecule, and making it from scratch requires very specific chemical conditions. The nucleotides that make up RNA don't spontaneously join together under normal conditions. They need help from enzymes or other catalysts. There was also the stability problem. RNA is much less stable than DNA, especially at high temperatures. The RNA molecules in an early RNA world would have been constantly breaking down and would need to be constantly replaced. For an RNA world to work, the rate of RNA formation would have to be faster than the rate of RNA destruction. Despite these challenges, scientists continued to find evidence that supported the RNA world hypothesis. They discovered that some of the most ancient and essential parts of modern cells, like the ribosome, which makes proteins, are based on RNA rather than proteins. This suggested that RNA-based catalysis came first in evolution, and that proteins were a later innovation. Scientists also began to find ways that RNA nucleotides might have formed naturally on early Earth. They showed that some of the chemical reactions needed to make RNA could occur in environments like hydrothermal vents or under the influence of certain minerals. While the complete pathway from simple chemicals to RNA remained unclear, it began to seem more plausible. The RNA world hypothesis represented a major shift in how scientists thought about the origin of life. Instead of looking for a single moment when life suddenly appeared, they began to think about evolution as a process that started with simple self-replicating molecules and gradually became more complex over time. This idea opened up new avenues for research and new ways of thinking about what the first life might have looked like. It suggested that life began not with cells as we know them, but with simple chemical systems that could reproduce and evolve. The RNA world became a bridge between the lifeless chemistry of early Earth and the complex biology we see today.